I would like to introduce Dr. Bruce Robinson. Dr. Robinson is an endocrinologist and head of the Cancer Genetics Laboratory and the Koling Institute at Royal North Shore Hospital. He was appointed acting dean of the Faculty of Medicine at the University of Sydney in April of 2006 and dean in March 2007. Dr. Robinson graduated from the University of Sydney in 1980 and then undertook studies for a Master of Science degree. His further molecular research work was performed at Brigham and Women's Hospital and Children's Hospital, Harvard Medical School from 1986 to 1989. He was awarded a Doctorate of Medicine from the University of Sydney in 1990. He has developed and led Cancer Genetics Laboratory since 1990 and has supervised over 20 doctoral and master's students working on genetic basis for tumor information and gene therapy. He has published over 160 peer-reviewed scientific articles. In 2003, Dr. Robinson was awarded the Daiichi Prize by the Asia and Oceania Thyroid Association for his work on pathogenesis and thyroid cancer. It is our pleasure to welcome Dr. Bruce G. Robinson.
Thank you, Karen, and thank you very much to those who are involved in organising this meeting for inviting me to talk. Now, that's actually not the right talk, I think. Anyhow, we'll press on. No, I think we've actually loaded the wrong talk on. Can we just pause for 1 minute? Okay, so we're all set. What I'd like to spend a little bit of time this afternoon talking to you about is some of the insights that we've learned regarding pheochromocytoma and paraganglioma from the work that we did when we set up the International SDH Consortium a couple of years ago. Next one. Okay, so what I'd just like to do is cover some of the same ground as we've already covered this morning, so I'll do that quickly, but then mainly spend time reviewing some of the findings from the consortium, particularly the patterns of inheritance, and then finally spend some time talking about recommendations for screening for affected patients and their relatives. I put up this slide just to emphasise something that's been touched on by many of the speakers this morning, and that is that taking of a very careful history is absolutely critical in these conditions. And I'm going to be focusing on the familial forms of pheochromocytoma and paraganglioma, but all too often we find that medical school students fail to take an adequate family history. And as you've heard this morning, many times the family history does give a good clue as to what's going on in these patients. In fact, medical history suggests that the war that was going on between the Hatfields and the McCoys in the Appalachian Mountains was adrenaline-fueled, and in fact that the Hatfields were affected with von Hippel-Lindau disease. As we've also heard this morning, there are at least 4 and possibly even a 5th familial condition in which pheochromocytoma occurs. MEN2, VHL, NF1, and more recently described the pheochromocytoma paraganglioma syndromes. And this slide you've also seen this morning too, and I think it's an important one because it emphasises the anatomical relationship of the paragangliomas to what I think is becoming increasingly clear as mutations in the SDHD gene. And this does show the anatomical sites of these tumours, as you've heard this morning, mainly in the head and neck region, but also down into the aorticopulmonary window area. In contrast, pheochromocytomas, or functional paragangliomas, as we've also heard this morning as they're called, arise in sympathetic associated paraganglia of the adrenal medulla, but also in these other extra adrenal sites like the superior cervical ganglion, and down here besides the spinal cord, right down to the organs of Zuckerkandl, and as you've also heard this morning, around the urinary bladder. Now, I think important piece of other history to emphasise to you as well is this very, very important piece of molecular sleuthing that was undertaken by Bora Basal in Pittsburgh. What he recognised was that chronic hypoxic stimulation, which occurred in people who lived at high altitude, led to enlargement and cellular hyperplasia of the carotid body. He hypothesised that that hypoxia would result in increased levels of hypoxia-inducible factor, increased levels of vascular endothelial growth factor, and thereafter vascular growth and proliferation of cells in the carotid body. What he noticed, and I suppose the most important aspect of this observation, was that he noticed similarity between that carotid body hyperplasia in people who lived at high altitude and the pathology of paragangliomas or carotid body tumours. And he therefore sat down and looked on his computer and evaluated genes in the 11q23 region, which was the region shown from previous linkage studies as being linked to the familial forms of paraganglioma. He looked in this region of chromosome 11q23 for genes that were involved in oxygen sensing. And lo and behold, found that there was a gene which encoded the D subunit of the succinate dehydrogenase enzyme, and thought therefore that this might be a good potential candidate gene for this disorder. He then, of course, did the usual thing and went on and looked and found mutations in this subunit in familial paragangliomas, mainly in a number of Dutch kindreds. This is not only a really important piece of intuitive science, but was the first time that there had been germline mutations identified in a nuclear encoded gene which encoded for an enzyme which existed in the mitochondria, as we've heard this morning, the energy powerhouses of the cell. And this is just to emphasise, as you heard again this morning, that succinate dehydrogenase is an enzyme. That is, it's a protein molecule which converts one molecule to another, and it does so in a part of an element of the body called the Krebs cycle. And it converts the molecule succinate to fumarate. Now, we know that it's made up of 4 subunits, A, B, C and D. And certainly B, C and D have been associated with familial paraganglioma. We also know that mutations in the succinate dehydrogenase A subunit importantly are associated with a condition called Leigh syndrome, which is where people develop, particularly children, develop inability to sense hypoxia due to an abnormality in their brainstem, and this often leads to their death at a very early age. So this enzyme clearly has a role in oxygen sensing and the generation of energy, or the main energy molecule in the body, ATP. Now, at that stage, we knew that there were mutations likely to occur within this gene, but we didn't really know a lot about the way in which those mutations would actually manifest themselves in patients. And so it was with that end in mind that we decided to set up the International SDH Consortium back in 2005. And we approached a number of centres around the world. We had wonderful cooperation from our colleagues in France, who supplied well over half of the 62 families that we were able to gather together for this consortium. But we also had good collaboration from people in New Zealand, in Scotland, Germany, where else am I looking at here? Canada, and Germany as well. So there were a number of countries from around the world who contributed families to this international consortium. The aim of the consortium was to attempt to determine whether there were genotype-phenotype correlations, first of all. That is, were there specific patterns of disease that were associated with certain mutations in these B and D subunit genes? And would that necessarily enable us to determine the course of disease for a patient with a particular mutation? The second thing we wanted to try to determine was whether there was penetrance patterns which would assist us in counselling patients and their families with this disease. And as I've said earlier, these were the countries that participated. This is really the first results slide, and it's a schematic of the SDH-D gene. which you can see here is made up of 4 exons. These are the, if you like, paragraphs within the gene which contain the genetic message encoding the protein that makes up SDHD. And each of these boxes here represents a family with pheochromocytoma or paraganglioma. And the corners of the box are filled in depending upon the manifestations of the particular disease in this family. And the first box to look at is the little one, which exists just here and here, below each of the main squares. And that red box filled in is indicative of the presence of malignancy in that family. And you can see that 2 of the families with SDHD mutations had malignancy. So malignancy is not a common feature of families with SDHD mutations. You can see also in the top right box, the yellow box, is where a head and neck paraganglioma is present. And you can see that virtually all of these families, bar this one here, have a head and neck paraganglioma. You can see in the bottom left box, the purple box, these are extraadrenal, I can't even read it, these are the abdominal ones, I think, the extraadrenal abdominal paragangliomas, and in the bottom right one are the extraadrenal thoracic paragangliomas. So abdominal and thoracic tumours can occur in families with SDHD mutation, but clearly head and neck mutations, head and neck tumours are far more common, and malignancy is relatively rare. Contrast this now with the results of the mutation screening in the families with SDHB mutations. The SDHB gene is one which contains 8 exons, so it's a much bigger gene, much more difficult to screen than SDHD.